now question number 5. Question number 5 is okay. It's about ecology and fish. So there is we need to okay, we have learned this that when some animal is being hunted or being bred or breeded, so what we need to keep a check, right? So this is all about that, that if we are fishing or hunting it, then we have to keep a check on it. So first of all, what is meant by sustainable resource? Exactly, how would you define it? Do not run out in the longer term. Okay, what else can we write? This is your point number 1. Now what can you do for point number 2? Okay, we can also write that the production is equal to their removal. So what should we write? The production is as rapid as their removal. Okay. Okay, the government of a country decided to set a fishing quota between 2012 to 2020. Okay, but if you look at the herring quota has been decreased. You can fish more in 2012 and 14 but less in 2020. Now use the information and describe the trend. Now description means you have to tell what is happening in what year. And then you have to suggest the explanation as well. Like why have you done so? Okay. Yes, yes. We have to describe and then give the reason. Now it's a 4 mark. It's a 4 mark question, so there is no need to write description in very detail. You can just say that the overall okay, overall there is decrease in size of no, because it's of like 2 mark of description and 2 mark of the explanation. There is decrease in size of herring quota. Okay, and what do you think about 2020? I guess it's a close season, right? Okay, or if you want to give value, what, because they have given description, so I would suggest that you should write the number. Just give a simple number that from 2012 to 2020 the quota size has been decreased from, let's say, 34 no 33,000 to 33,000 to 2,000, right? That is from 33,000 herring quota to uh, what we have written, 2,000 herring quota. Okay, there's no need to give a value for like each year. Okay, why do you think so? Why the value has or why the quota has been decreased? We have used some word in the start of this one. It's not sustainable. Yes, it's not sustainable. Exactly, exactly. It's not sustainable. It's they are not like produced, or there can be overfishing as well, right? Too many caught, good. Too many caught in the breeding season. I guess this is all. Okay, let's move to the next part of this one. The next question is okay, how to conserve it, about the conservation of it. Okay, what can be explained two other named methods that can be used to conserve fish stock? One is quota system, what other things can be done? There should be protected areas where it shouldn't be allowed. Okay, what else can be? Net size. Okay, net size are not caught. So baby fish or smaller fish are not caught in the net. Okay, what is the other method? Fishing, not breeding, fishing. Uh, no fishing in breeding seasons, right? In breeding seasons. I'm also as out as you are. I've also taken back to back lessons today. Okay, next question is okay, fish are a good source of lipids. We know very well that a fish oil is really, really good for our health. Is it so? Have you ever taken the fish oil capsules? No? At my age, we need to take it. Okay, it's a 5 mark question. The question is fish are a good source of lipids in the form of fish oil. A student investigated the digestion of fish oil in the laboratory. A student set up an experiment. Okay, because okay, why 8? Because the enzyme for the oil one act like really well at 8 pH. Okay, the lipase one. We know that lipase work in the alkaline environment in this stuff in this sorry, small intestine. That's why we've given 8 pH, and we have attached the pH meter so that the pH won't be disturbed. Okay, sometimes we also develop a buffer. Buffer mean that if you add a certain acid or a certain alkali, it won't change, the pH won't affect it. Okay, that is buffer. Okay, the student recorded the pH of the content of each test tube using a digital pH meter at the start of experiment. Both meters record a pH of 8. The student continued to monitor the pH for 30 minutes. Use your knowledge of how lipids are digested to explain what would you expect to happen in each tube. Okay, here we have the bile solution and fish oil. But here we have fish oil, bile solution and lipase. So first of all, let's discuss the function of bile. What is the function of bile? Yes, neutralizes the acid. And the second function is break those bigger fat molecules into the smaller one, emulsify fats. Okay, what is the function of lipase? Into fatty acid and glycerol. Okay, I forgot to mention one thing. I was about to tell you very important something, really important. Totally skipped out of my mind now. Okay, what is the purpose of emulsifying fats? Good. To increase the surface area so that enzymes can easily act or act on them from like all sides, from any side. Okay, use your knowledge of how lipids are digested to explain what happened in experiment 1. Now here there is no lipase. So what would happen in 1 and what would happen in 2? Only emulsification has taken place in test tube 1. right? Okay, if you want to write it for 2 mark, what other thing you can do? No digestion take place as there is no lipase. Okay, what would happen in experiment number 2? Hmm. And what about pH? You have to mention the pH as well. So, okay, here the pH remains same. Okay, now I'm telling you the reason. Remains same. Why? Because fats are not broken down, right? They only emulsified. Okay, here the pH would lower. Why? Because here we have fatty acids, right? Fatty acids by the breakdown of fats by lipase. Okay, so we have to describe all the like points that were given. Okay, let's move to the next question.